you for inviting me. It's a really great group to talk to. You'll have this great seminar series. It's pretty amazing. I'm kind of embarrassed to be part of it. But what I'd like to do today is talk to you a little bit about the evolution of genome structure, primarily in Drosophila. I gave it this general title because we've been moving into studying a lot more species and different genomes, but you know, make no mistake, today is going to be mostly a fly talk. And so this is the short title, Variation in Genome Structure in Flies. And if anybody feels like tweeting or whatever it is, I don't care. You go ahead and do it. I don't need to worry about keeping this you know, confidential or anything. One thing I would like to say, again, I think we're running into the, there we go. One thing I do want to say is that this theme will be about these three things: hidden, heterogeneous, and functional. So this is going to be the theme. Unlike a lot of evolutionary talks, especially some of the ones I've given in the past, I'm going to be a little bit light on the technical detail and even some of the theoretical conclusions, because I think this is the beginning of a long journey where we're having to come to grips with the fact that a lot of this variation is hard to find and it's very heterogeneous. And so we need really good methods to do this. So I'm going to sort of take you on a tour of what we've been doing for the last couple of years in this direction. Okay, so I'm going to start my talk with the standard biologist or evolutionary biologist diversity slide. And I like to show this slide because it reminds me that I'm fundamentally interested in the origins of genetic diversity that lead to phenotypic diversity in various organisms. For example, the diversity that we see in closely related species like these strawberry poison frogs, or given enough time, all the amazing helmet morphology that you can get in these membracid beetles here in the middle. And I apologize if you can't see my pointer, but if you, and of course, one of the most interesting types of genetic diversity that we'll often see are the phenotypes that have led to, or the genotypes that have led to huge phenotypic diversity in domesticated animals, so famously pushed by Darwin. And one of the most extreme examples of this is in dogs. This example in the upper left-hand corner is a example of me using a Bing image search. Yeah, first time anybody uses Bing, right? But a Bing image search on the term dog. And I've only taken a screenshot from Basset to Boxer. You can see there's huge amounts of diversity in coat color, height, shape, size, temperament, and everything like that. And of course, as well, this is also a lot of diversity in our favorite, in relatives of our favorite organisms, Drosophila. So these are the Hawaiian Drosophila showing their amazing wing morphology. Okay. So what do I mean when I talk about genetic, whoa, something, yeah, there's a little technical detail going on here. I'm not sure what's going on. Pardon me, sorry. My computer is lagging a little bit for some reason. I'm not sure why. Okay, I will figure this out. There we go. Okay, so whenever we talk about genetic diversity, what is it that I mean, or genetic variation? Well, whenever you talk to someone who's not an evolutionary biologist, one of the first things that you'll often hear is, well, humans and chimpanzees are less than 2% different than one another, right? And so we'll go back and say, oh yeah, the difference in humans and chimpanzees is 1.2%, and maybe the difference between gorillas is 1.6%. Okay. And what we're typically talking about when we talk about that kind of genetic diversity is you have about, you'll take well-aligned regions of the genome, or well-conserved regions of the genome, you'll line them up and compare the number of differences in nucleotide alignment, and that's what you get here. If we do the same exercise with Drosophila, we can say take Drosophila melanogaster and compare it to Drosophila simulants, and we get a similar result, around 4.5%. This number varies depending on which part of the genome you choose and what kinds of alignments you do, but roughly speaking, you get about 4.5% difference. But I'd like to step back from this perspective of genetic variation and think about different types of genetic variation. So here I'm going to take my diversity slide and take a slight angle on it and start looking at types of variation that you might think about in terms of their direct genetic causes. So here I'll talk about dog height, industrial melanism, the famous bar mutation in Drosophila, insecticide resistance, and some interesting corn phenotype. So this is one of the most striking examples of dog morphology, is that you have these small dogs and these large dogs. And it turns out that the dogs on the top lack a retrogene of the fibroblast growth factor 4. In fact, there's two of them. The middle row of dogs is actually for a duplication onto chromosome 18, and the bottom row of dogs is from a duplication onto chromosome 12. And they also have serious genetic diseases as a result of that. But moving on, industrial melanism is this famous example. And back in 2016, there was a paper that was published that said this variation in melanism between these moths in the UK have this genetic variation that's due to a transposable element that jumps into the first intron of the gene cortex. And so you have a mobile piece of DNA that's jumping into another, into a region of the genome and causing phenotypic variation that's relevant to adaptation. We have now moved on to, again, back to one of my favorite organisms, is to give another example of a type of structural variation that's very important and near and dear to my own heart, which is duplication. So this is an example of probably, let's say, maybe the very first copy number variant that has ever been characterized, which is the bar mutation in the Drosophila I, which, depending on how many copies you have due to unequal crossing over, you can get a phenotype that is severe to less severe, okay? Next slide. And then another important phenotype is this ubiquitous resistance to toxins, especially insecticides, that many insects all over the world have. It's an especially important problem with crop pests and disease vectors. And there's a famous example in both mosquitoes and Drosophila of CYP6G1, the cytochrome P450, that in Drosophila has an amazing situation.
I think we need the next slide.
Oh, next slide, please. Yeah. Next slide. The CYP6G1 mutation, where you have a duplication of the gene plus an insertion of several transposable elements both before and after the duplication, leading to both duplicated transposable elements and single copy transposable elements. And what this mutation does is it causes a fly that bears some of these derived alleles to be more resistant to, for example, DDT, or in examples I can show you later, they'll be resistant to caffeine or nicotine or something, or other toxins like that. Next slide. Another interesting mutation, which is also a duplication, is this duplication of the ZM19 Madsbox gene in corn. And this leads to this interesting phenotype called pod corn, where these leaves encase each of the kernels, right? And so each of these mutations that I've shown above, although some of them are under domestication, are spontaneous mutations that you see if you're a breeder or you just look in the wild. Next slide, please. So I hope that I've communicated that structural variation is important. And very soon I'm going to show you just how common we've been finding it in fly genomes. We've known that structural variation is common for a while, but I'm going to show you, I think it's even more common than we thought before. And I hope I've also communicated what we're talking about when I'm talking about structural variation. We're talking about rearrangements of the information of the chromosome. So duplications, deletions, insertions, rearrangements, things like that, that change the information structure rather than changing the spellings, let's say. Okay. Now today, next slide, please. Today, I'm not going to spend a lot of time on technical details about assembly, but I do want to show you that high quality assembly, genome assembly is possible. Next slide. And we are doing this with long read sequencing using single molecules, primarily with PacBio, Pacific Biosciences, but also with Oxford Nanopore. And here's just two examples of how we're doing this. It's very, very important to start with high quality DNA. So if we look at the left-hand side of this figure, we extract high molecular weight DNA. And this is the one step you absolutely cannot skimp on. And so if you want to take away anything technical from this talk, is that you can redo the assemblies all day long and make it better and better. But if you don't get good data, you will not get a good assembly. So if you need to take anything away, Get out your pulse-filled gel electrophoresis, be very careful, have wide mouth pipettes and things like that. The approach that we're primarily going to look at is this meta-assembly approach, which we use quite a bit, where we'll take our data from a sequencing run, and then we'll make 2 assemblies, and then we'll merge them and use the complementary benefits of each assembly. And if we'll go to the next slide, you can see that this works both with 2 different assemblies from the same data or 2 different assemblies from different data. And so we've made use of this pretty extensively in our work. Next slide. And this is a slide that I present whenever I'm speaking about mosquitoes, and it's just a brief display of the contiguity of these assemblies. So how many gaps are there? How long are the individual contigs? And so on the x-axis, you have the contig in 50, which is the length of the middle contig. In other words, the contig where any contig bigger than that is half of the genome if you sum up all the contigs. And the left is the y-axis is the scaffold in 50, which is just what happens when you chain those contigs together into the appropriate order and orientation. And this is just a way to say that the closer to the upper right-hand corner you are, the better you're doing at assembling your genome. And just for reference, there's a lot of different mosquito genomes on there just to show you what's possible and what happens. As you'll see, there's a lot in the left-hand side and lower left-hand corner. Next slide, please. And I'm going to focus in on this area because this is what concerns us. So next slide. Perfect. And what we have here is the big square box is one of the genomes I'll be talking to you about today. That's Drosophila pseudoobscura. So it's got one of the most contiguous assemblies in all of Drosophila. And then on that cluster on the bottom left is all the Drosophila melanogaster genomes. The fact that they're both have a contig in 50 and scaffold in 50 are nearly the same, that's the important part. Bigger genomes with bigger chromosomes can get farther up and to the right, but the fact is that they're quite close to the diagonal, and that's important. So if you push the next slide. And again, this little polygon here is the Drosophila melanogaster species complex, including polymorphism and divergence. We'll see in a moment. And this plus sign, next slide, is Drosophila pseudoobscura. Okay, so with that out of the way, we'll move on to the next slide. These assemblies that I'm about to show you, I don't have time to go into the detail about how we calculate all this, but we have a lot of methods. Please feel free to ask me. Our worst assembly for today's presentation is 1 error per 25,000 base pairs, and it goes down as low as 1 error per 150,000 base pairs in Drosophila pseudoobscura. For those who keep track of these things, it's a Phred score of 44 to 52 range, and more than 98% of the genome is complete, which is comparable to Drosophila melanogaster's reference assembly. So we're using the Busco's metric to determine that. So next slide, please. So if you're interested in some of these methods, the primary merging method is described here in the next slide. And some applications to this are also discussed for Oxford nanopore in this manuscript. Okay, so if we can go on to the next slide. Finally, a huge part of this, of the results I'll be giving are actually how to determine where these mutations are, these structural variants. We have 2 pipelines to do this, and we're continuing to refine these approaches, but you can find them on GitHub at these 2 locations. So next slide. And with that, I'd like to close the sort of the approach section of the talk with a quote from John Gillespie's book on population genetics from 1996, which talks about Marty Kreitman's 1983 paper, possible. In 1996, it was possible just 13 years after that paper to an undergrad to do the same project, which took Marty Kreitman years to do and lots of danger and lots of time and very difficult. Now I'd like to say that that same thing is true, except now you can sequence a polymorphism data set for many different reference genomes and study structural variations. This is not hyperbole. Undergraduates in my lab have sequenced entire genomes, and we're now doing a paper on that using Oxford nanopore. Okay, so next slide, please. Finally, we're going to actually move into this into the meat of the talk. I apologize that we've taken so long to get here. I think we're going smoothly now. So if we move on, we're going to go to the flies. And start thinking about structural variation with Drosophila melanogaster. To start off, I'm going to talk about polymorphism data sets from 2 projects. So if we'll move to the next slide. Some of the results described here can be found in this paper, published a few years ago with the lead author Mehul Chakraborty, and then the next slide. Another study that expanded to a large polymorphism data set done in collaboration with Tony Long and Stuart McDonald on the Drosophila synthetic population resource, which is a great QTL mapping resource that enables us to do a lot of really cool things. So if we'll move on to the next slide, you can see that this Drosophila synthetic population resource is a QTL mapping resource that has a lot of parental lines that are used to create a recombinant inbred panel. And I'm going to focus on the parents, not the recombinant inbred panel. And they're drawn from all over the world to sample diversity of different regions of the world. And whenever we're looking at the entire population set, that will be the collaboration with Tony. And if we go to the next slide, whenever we're looking at only a pairwise comparison, we're talking about a comparison to this Drosophila A4 strain and the reference genome that we all know and love. So if we move on to the next slide, if we look at just A4 and compare it to the reference genome, you can see a lot of variation immediately. This is a circus plot where each ring represents a type of variation. The inner ring are types of variants that are structural variants that are not transposable elements or CNVs, and they're just indels. And you'll notice that they're basically ubiquitous across the genome and distributed everywhere. The same thing for CNVs. When you get to the green and then the outer purple ring, you'll see the same thing for transposable elements. And the biggest thing to see here is that, and we'll show you this later, is that variation is everywhere. And the amount of variation we're seeing here is a lot more than we've ever seen before. Okay, so if we go on to the next slide, we're going to focus here on transposable elements. Now, this is a similar plot to before. The only difference is that I varied the window sizes so that you can see the density doesn't depend on window size, because there's some weird window size type of dependency. And what you can notice is that there's a couple of hot spots where you see very bright yellow streaks and a couple of cold spots where you see some purple streaks. But by and large, you just see that the entire rings are just lighting up. There are TEs all over the place. They're littered across the genome, and actually you're missing a lot of them. If we go into the next slide, we'll see the same thing for what happens instead of insertions of transposable elements. You get duplicated regions of the genome. In this particular example, you'll see a couple of notable very hot spots. Also, you'll see a lot less variation, but one thing you will see is that the X chromosome has a lot more duplications. This is something we've seen before, but now we can find almost all the duplications that exist in the euchromatin, and it's a very clear signal that the X chromosome is enriched for CNVs. So if we move on to the next slide, we can see a summary of what we just described. So on the red bar on the left is non-structural variation. So these are SNPs, small indels, and such, and that affects upwards of 2 megabases in the region of the euchromatin we assayed. On the other hand, if you look at the dark blue bar immediately next to that red bar, that's the amount of genetic variation due to structural variation that we could not see from Illumina-based methods, right? And you'll notice that there's more than twice as much variation in that category than we have all of non-SV variation. Of course, we could see quite a bit of structural variation before we even got to PacBio and long-read sequencing, and that's the very large light blue bar. But it is nice to be able to detect most everything with one single platform. Okay, next slide. Okay, and so, okay, so that's not quite coming through, but this is and I'd like to say that our observations of missing, wait a minute. So, do you not see this orange bar plot in the previous slide?
There's only the text.
Okay, so we just lost a slide, but that's okay. So the previous slide was just looking at, well, how are we missing genetic variation? So what types of variants are we missing? Well, it turns out that things like inversions are really hard to detect, right? We miss about 80% or more of inversions. Also, if we look at copy number variants, we're missing about 60% of them, right? And as there are other types of variations like non-TE variants, which we mostly get without PacBio approaches. In TEs, we missed maybe about 30%. So that was just a summary of the kind of variation we're missing. And this paper recently came out. It doesn't have the title on here, and this title just says Haplotype Resolved Diverse Humans, an Integrated Analysis of Structural Variation. And this is a paper released from Evan Eichler's lab that basically, yet again, reaffirms some of the things that he's found in the past and our lab has found in the past, is that we're missing a huge amount of structural variation whenever we were using previous methods. Okay, let's move on to the next slide. Oh, yeah, there you go. And so the question I'd like to ask is, if I were to pick a random gene in the genome in a diploid in a fly, how likely is there to be a structural variant that's affecting that gene? So this is what this plot shows. On the x-axis is the percentage of genes in a particular size category indicated on the y-axis that are burdened by at least one structural variant. And we did this through randomly pairing our haploid genotypes through all possible combinations and looking at the distribution. And what you'll see is that if you just pick out a random small gene, you'll find a couple of percent, 2-3% of the genome of genes have a structural variant in a single fly you pull out. This is not a polymorphism sample with segregating variation at some low frequency. This is in any single fly you pull out, a couple of percent of the genes are going to have at least one structural variant. If you keep making the gene larger and larger, the target size gets larger. And let's say a 10 kb gene, you'll have roughly 30% of genes will have some sort of structural variant segregating if the sample that we're using is representative. Next slide, please. If you focus on those genes that contain structural variants, so this is conditional on structural variation, how many structural variants does a gene contain? This plot is the same as before, but now the bars represent the share of one copy in purple, ranging up to more than one variant in purple up to more than 5 in yellow. And as you can see, you don't have to have a gene that's very big to have 2, 3, 4, even more structural variants in the same gene. So these very rare ultra long genes in Drosophila will have lots of transposable elements jumping inside their introns. Okay, next slide, please. So the question is, what is the importance of structural variation for phenotype? Well, we're really lucky that this Drosophila synthetic population resource exists because there's been several QTL studies that have been done, and this is before we actually had long read sequencing. So what they did in these QTL studies, very typical, is that they will go into the peak where they think the variation is, and they'll look at the genes, and then they'll put a candidate list of genes that they think are worth following up. And so we took a list of all of those genes and looking to exclude any genes that were picked because they were known to be structurally variable. None were actually that way. But we summarized across all possible studies. And if we go to the next slide, we summarized across all possible studies, and we expect randomly that you would expect to see about 11 genes would have a structural variant inside their structure. And we actually observed 16. Now, this is not a large number of genes, and the number of studies is relatively small, but this represents years of work integrated over multiple groups. So it's like really hard to get this data. But if you go to the next slide, you'll see that this is 45% enrichment. Now this is marginally significant. I can't tell you it's not significant, but I'm not super confident about this. I just think we need to do more work. But this is a nice suggestive idea. If I could increase the sample size, I would. But this is the cards that we were dealt. So if you've got another 10 studies and another couple of dozen genes or 100 genes, then maybe we can increase the sample size on this. Okay, so let's move on to the next slide. In the next one. So now we're going to start looking at classic population genetic signatures. So the first thing that we're going to look at here are these what's called the site frequency spectrum, which is an allele, which is the histogram of allele frequencies. And first, I want you to focus on this yellow synonymous category here. What we have here is that singletons, doubletons, and tripletons, so one copy are present in one individual in the sample, present in 2, present in 3, etc. I want you to notice that you have this nice shape. And what's indicative is if you have an increase in the singleton, that typically means that relative to the synonymous categories, that typically means that these particular mutations are under purifying selection. So as you might expect, mutations that change the amino acid sequence in the sort of pink color, those have more singletons, and those that are duplications have even more singletons, and TEs have even more singletons. And as you'll notice, the low frequency, the frequency gets lower and lower as you move to the right, and this is an indication of strong purifying selection. So if we move on and just focus on SVs, this is a later study where we're actually able to unfold the site frequency spectrum. The idea is the same. If we start off with the duplications, we'll notice, which is in brown, we'll notice that very, very, very few mutations get to high frequency. There are some, and TEs are at extremely, extremely low frequency. In fact, mostly singletons. Let's move on to the next slide. Okay, so now these are genome-wide patterns. I hope I've convinced you that that we're missing a lot of the variation, and that means that variation, even though it was ubiquitous before, it's even more ubiquitous than we thought. And also that it's strongly under purifying selection, and that these mutations can have a strong impact. But also it seems like some of the variation affects phenotypes. And those phenotypes, they may affect them in a negative way, but they seem to often be enriched in mutations or in QTL peaks. So let's go ahead and look at a couple of examples. This is a region where I am comparing the genotype of ISO1, which is a reference strain on the top, to the strain that we sequenced on the bottom. And you'll notice that there's a missing gene in the middle, the CYP6A17. This is a cytochrome P450. Next slide. And what you'll notice is that that whenever we do what every self-respecting fly biologist does, when we find an interesting mutation, we go to Flybase and see if anybody's published on this. And so we plugged this into Flybase and we saw, wow, this gene CYP6A17 is required for temperature preference behavior in Drosophila. And so what we did is, next slide, is we decided to test this ourselves. We replicated the setup behind that paper, which did genetic manipulation of that gene and knocked it out and found out that it has this phenotype. So we decided to determine, does this phenotype exist in the wild? So we took this setup, which I engineered in the lab on our floor, and set up a temperature gradient with ice on the left and hot water on the right, and an aluminum bar connecting these two in the middle, and a microcontroller to where we can measure the temperature gradient. And then we put flies on a little stage, next slide please, where they could choose their preference. And one lane is for one genotype and one lane is for the other genotype. And you can see that they can array themselves out on this stage to their preference. And we do this in the dark and then take a photo after the behavior is over. Next slide, please. And if you do this and randomize the lanes and you do 6 replicates, you'll find every single time that the A4 strain, which is missing this mutation, are much less averse to cold than the strain that we use, white 1118, which is exactly what they found in the genetic study. So that genetic study is a really good example because they control the genetic background. But here we see that it's basically a Mendelian mutation. Okay, so if you move onto the next slide. This is one of my favorite examples. So I'm just sort of marching through some of the interesting examples that we found. So if you'll see on the top, we'll see ISO1 has one gene, CYP28D1, but on the bottom, you'll see CYP28D1 proximal and CYP28D1 distal. So you'll have 2 of these. So next slide. And again, we go into the Flybase literature and look up CYP28D1, and what we find is that the most interesting hit in Flybase is from our own floor, which we were not expecting. Tony Long, if you'll see down here, find that nicotine resistance in Drosophila. And so this is where we were just blindly looking at interesting genes and we found this mutation. And what you'll notice is that between these 2 copies, you'll see this little pink and blue thing, which is an accord fragment, which is a type of LTR retrotransposon. So if you move to the next slide. So we decided to see what are the consequences of this. So what we have here are comparing a strain containing the mutation A4 to one that doesn't have the mutation A3. And we'll notice that A4 under normal conditions has 2 copies, the light and the dark blue, and it's expressed at a much, much higher level. Next slide. And also the genes flanking it, the CG7742 and CYP28D2, and are much more highly expressed in A4, but not in the strain that lacks the duplication and the accord fragment A3. If you go on to the next slide, you'll see that under nicotine conditions, A3 does in fact induce the cytochrome P450, next slide, but it does not induce the surrounding genes. So what we think is going on here is that you have this accord fragment, which is upregulating the entire region, but the duplication is also contributing to the increase because there's 2 copies to upregulate. So next slide. And if you look at genetic variation surrounding this region and you look at these markers for a skew in the site frequency spectrum indicative of recent selection, you'll find these really, really high peaks, which are very, very significant surrounding this region. And the green span is all mutations we've ever discovered in this region for CYP28D1, and the blue span is for this particular study. And you'll notice that I don't know how to do this correctly because we're dealing with regions of the genome that aren't homologous, so I'm not sure how reliable this is, but there's a fairly strong indication that this is something that's been under selection. Next slide, please. And finally, if we open this up to an entire polymorphism sample, you find that in this middle panel, third from the bottom, you see this A4. That's the mutation that we just characterized, but there are several different haplotypes. Anytime you see a triangle, that's a transposable element. Anytime you see a box, that's a duplication. You can see transposable elements co-occurring with duplications and triplications and things like that. So this area is extremely allelically heterogeneous. So you can't just count on having one single mutation. Next slide. And just to refer back, this famous CYP6G1 mutation that I talked about earlier has duplications and transposable elements inserted into it. And this is well understood and has been published on, especially this paper from Schmidt et al. From Charlie Robin's lab back in 2010. It's a great paper. And you'll see that that's very allelically heterogeneous. Next slide, please. But we just sampled more and we found 3 more alleles. We weren't even trying to find this, but again, the allelic heterogeneity is just crazy for some of these regions. Next slide. And finally, I'd like to say it's not all the cytochrome P450s. Okay, so the temperature preference cytochrome P450 isn't involved in toxin response, but it's still a cytochrome. Things like insulin receptors are also extremely allelically heterogeneous. Each of these triangles represents a transposable element insertion into this gene, or the little red gaps represent a region where there's a TE in the reference genome. And what you'll see here is that we have, for every single strain has a slightly different, pictured here, every single line has a different haplotype with a different TE insertion profile. Okay, next slide. And what's interesting about this is that you have these TEs are inserting in and near transcriptional regulators where you have these transcription factors and marks of active transcription have TEs immediately near them. So there's quite a possibility that these transposable elements are going to affect the expression of this gene. Okay, next slide. Okay, so now we are moving into, and I apologize, I wanted to be here a little bit earlier. Again, I think I'm missing quite a bit of time due to my technical difficulties. Now I'd like to go into our recent between genome species comparisons, and here we're going to be talking about the close relatives, Drosophila, Melanogaster, and its 3 most close relatives. So move on to the next slide, please. And of course, in that previous slide, I really want to acknowledge this is a huge collaborative effort with all the folks mentioned on that page: Amanda, Larry, Quinte, Jinghua, Zhang. I'm not going to say everyone's name: Colin and Colin, Michael, John, and Christy Montooth, and Jeff, Adrian, and Jeff Vidinayagam, and Danny Coast. I don't want anybody to think that we just did this in our lab. This is a big collaboration. Okay, next slide, please. And Colin, Michael, John. Okay, so if we, this is the Drosophila phylogeny of the most close relatives to Drosophila melanogaster. If you go to the next slide, we're going to focus in the next couple of slides on first this top region, the Drosophila Simulin species complex, and its sister species Melanogaster, and then we'll move on to Drosophila pseudoobscura. So if we go into the next slide, I'll remind you earlier that so So in the red, we have this Drosophila simulans complex, or I'll just call it the Sim complex. And those are 3 species, which are effectively a trichotomy, probably incomplete lineage sorting. But you see Drosophila marciana, Drosophila seychellia, and Drosophila simulans. And then the entire group, if you include its sister species, Drosophila melanogaster, you have the Melanogaster species complex. And they're very closely related. The divergence between them has happened only in the last couple of million years, and the Sim complex is only diversified for less than 1 million years. So we go on to the next slide. And in this time, the difference between Drosophila melanogaster and the Simulants complex is less than 5%. It's about 5% divergence in 50 kb windows that you can align. And this was reported about 14 years ago now. And this is a, kind of a rough mental model of what people have when they think, what's the divergence between these species? So if you go on to the next slide, you'll see that what happens when you take these high quality reference grade assemblies and you align them across the entire genome, and then you count up how much of the genome aligns and how much of it doesn't. Well, first thing you'll notice is this green bar. We're comparing to this black line. The green bar is kind of what they did in that 2007 paper, except now we can align more of the genome. And so once you start aligning parts of the genome that are slightly harder to deal with, you'll find more and more differences. And so we do have an increase in the divergence there just a little bit, but not a large magnitude. But what's really interesting is that in the Chromatin and the regions that we think we've got really well assembled, we see upwards of 15% of the genome is either in duplicate or in TEs or involved in inversions or otherwise not alignable in a unique way to the other genome. So this concept of how much divergence do you have between species is really kind of colored by our history of thinking about single nucleotide and small indel substitutions. So there's a lot more variation than we think there is, and we should start thinking and talking in terms of this rather than just the single base pair substitution rates. Next slide, please. Oh goodness, this should, well, this is fine. Just a reminder that this is work done by Danny Coast and Amanda Laraquinte. Here we're looking at the chromosome 3L, which is an autosomal arm in Drosophila, and it is just looking at the repeat profile across the chromosome. The red vertical line on the right-hand side of each of these rows, and each row is a different species. The red line is where the heterochromatin border boundary is, and you start getting a lot more repeats. And you'll notice that you get this, typical signature of lots of transposable elements and repeats that start around the heterochromatin boundary. If you go to the next slide, and they're pretty similar across both species, or across all 4 species. But what's interesting is if you look at the X chromosome, you'll find that Drosophila marciana and Drosophila seychellia have a different profile on the X chromosome. So if you go ahead and go to the next slide, we'll zoom into the X chromosome, and you'll notice all of these little blue spikes all over the X chromosome. And you'll find that that's a huge increase in the number of satellites arrayed across the U chromatin throughout the X chromosome. So this is something that we previously had a very difficult time in studying because satellites are among the hardest things to do with short read sequencing. So you're left with hybridization methods to get a handle on how big these regions are. Next slide, please. So now I'm going to move on to kind of the last part of the talk for the Simulants clade, and just sort of propose this idea. Going back to some of the nice models that we think about in population genetics and with real mutations, we ask the question, with transposable elements and effective population size, does population size matter? And some of the earliest measurements in Drosophila going back to the early 80s and trying to quantify the number of TEs say that the number of TEs in Drosophila melanogaster are hugely much larger than those in Drosophila simulants. Some estimates were as much as 7 times higher, and they range anywhere from twice as high to 3 times as high up to 7 times as high. And those particular estimates have come as recently as the 12 Genomes paper. And so this got people thinking way back as soon as we could measure diversity in Drosophila simulans is that, well, if the population size in Drosophila simulans is greater than that in Drosophila melanogaster, and TEs are often deleterious, maybe this explains the fact that Drosophila simulans has a lot fewer TEs. maybe Drosophila simulans just has more effective purging of these bad mutations. Well, I think it's probably not that simple because the rest of the Simulants complex also has few TEs, but also because it seems like we've grossly overestimated, at least in the Chromatin, the amount of TEs that Melanogaster has in excess compared to, say, Drosophila simulans and the rest of the Simulants study. So next slide. So if this is just a comparison of genomes where each pair, starting from the left, just shows Drosophila simulans, the old amount of TEs versus the new amount of TEs marching on down. And what you can see here is that with our new, the three high bars that you see here are our new assemblies, and we just find more and more TEs. And so the amount of variation in TE content is a lot less than it used to be. So if we'll move on to the next slide, we can look at this across the entire Melanogaster complex. And on the top left-hand corner, you'll notice, yes, Drosophila melanogaster absolutely has more TEs, but the amount of excess it has compared to, say, Drosophila simulans and Drosophila seychellia is not that much, especially compared to sevenfold. It's not remotely sevenfold in the Chromatin. Now, if you go into this middle plot, what you'll see is we've inferred the TE content. These are not number of TEs. That's a much harder thing to do. These are just the number of bases that are annotated as TEs. And in this middle plot, we've sort of broken out the amount of TEs to where they've fallen out on the particular branches. And we'll find that most recently, the terminal branches, that these are all probably strongly enriched for polymorphic TEs or very, very recent TE fixations, and they're dominated by LTRs, long terminal repeat transposable elements. But in the Simulants complex, there are quite a bit of TEs that are from this, from the DNA transposons, both within 2 or 3 members of any individual. So you can see that in this red and the blue balls. However, if you go all the way back to the ancestor of the entire clade, you'll find a domination of non-LTR richer transposons. And if you want to see this in a relative manner, just look in the bottom right-hand corner. It's just rescaled. And I think it's important to note that these sorts of patterns have been found before. But one of the things that I just showed earlier is that we're not really certain about what exactly we're saying. Now, of course, some of the things that we've been talking about, the excess of TEs and things like that, not everybody said that. In fact, Robert Kofler and Christian Schlotterer are among people who got it quite right in their 2015 paper about the content of these TEs. And that's going to be a recurring theme. We're going to see that, Schlotterer gets things right earlier when it was harder pretty often. So if we'll move on to the next slide, we're going to take a quick brief segue to another type of tandem repeat, which are these tandem repeats involving tRNAs, these genes involved in translation, the template for loading up the amino acid for translation. And one of the things that we noticed when we first sequenced Drosophila melanogaster is that there's about 300 in a genome. However, whenever you look at Drosophila simulans, previous studies found only about 255 or 268. But we found 300. So again, this is one of these things where you can even have a whole genome consortium project to sequence the genome of Drosophila simulans, and you just don't get it right. It's really amazing what long read sequencing has done to improve our ability to detect these important functional elements of the genome. So this is another example of structural variation because these are tandem arrays that often vary in copy number. And so they do turn over quite a bit. And if you look at this plot, you can see a lot of how that turnover happens. But if we move on to the next slide, I'm going to come to, and I guess this is the second, this is part 2 of how Christians group and people here in Vienna get things right earlier than people working with long reads, is this Y chromosome study. So Y chromosomes are very repetitive, TE-rich, repeat-rich chromosomes. They're heterochromatic, and they're very difficult to study. So what we found is that once we assembled the Y chromosomes, and this is work done by Amanda Lerpente and Jinghe Zhang, is that there's evidence of duplication for every Y-linked gene except for one. So they're in multiple copies arrayed on the Y chromosome. And also they identified 41 imported genes from other chromosomes. And as I alluded to before, this had already been discovered earlier in 2017, if I'm not mistaken, by Tobler and Schlotterer, and they already reported this pattern, although it is useful to know that we're able to get a more comprehensive pattern and discover many mutations that they weren't able to discover. But that's only fair because using short reads is so much cheaper, especially back then. But it does underscore that even whenever people have found the patterns, getting them completely comprehensively right still requires things like long reads. Let's move on to the next slide. And finally, I want to get to one example. I like having a little example for each of these sections, and that's to give you some idea of what's actually going on a sort of genetic as well as phenotypic level. So in Drosophila simulans, there is this mutation, and I apologize, I'm going to go a little bit over the talk time just because of technical difficulties. Is that okay?
Sure, absolutely.
Okay. So we're pretty close to the end. I apologize. I think I rushed because of how much time we lost. But okay. So here we go. I'm looking at this region in the Drosophila Melanogaster X chromosome, which has this gene called maternal haploid. Okay. And I apologize. I'm going to have to open up my slide notes because there are some things I don't want to say wrong. But okay, so we're looking at this maternal haploid region. And maternal haploid is a gene that maintains genome integrity, and it interacts with the 359 base pair satellite repeats, and it's essential for proper distribution of parental genomes during zygotic cell division in Drosophila melanogaster. Okay, so it's an important gene that interacts with the repetitive genome, and it's important for cell division. Okay. We were quite surprised to see that this gene was actually duplicated in Drosophila simulans and Drosophila mauritiana, just a standard straight up tandem duplication. And what we found is that, well, you can see it right here, the transcript of the distal copy of maternal haploid in these species is shorter than the proximal, and the proximal retains a parental gene structure. Next slide, please. And what's fascinating is if you take RNA sequencing, also this isoseq data, FYI, is from Pierre Nahoud's data, again, another connection. But what you'll notice here is that you have RNA-seq data from females in the middle and RNA-seq data from males on the bottom. And you notice that here that there's a huge difference in the proximal and distal copy. You'll notice that the distal copy has a bias toward male expression, and the proximal copy has a bias toward female expression. And you might not be surprised that a gene called maternal haploid would have a female biased expression pattern. But what's curious is the expression pattern for males. So if you go to the next slide, we summarize this with a expression in 3 copies. You'll find that the proximal copy, so this is the ratio of the proximal to distal copy of the expression. So see that the proximal copy in females has a huge bias. So it's very enriched for females. This is just summarizing that previous slide and adding 1 tissue. And you'll find in both male and testes that it's exactly the other way around. So you find that these are specialized, with the proximal copy specialized toward female expression and the distal copy toward male expression. So we don't know what this does, and I think that there's a lot of good biologists out there who are probably interested in studying this, and so I think, have at it. So moving forward, next slide. Finally, I'm going to conclude with some of the things that I'm most excited about, because we finally started to integrate a lot of these genomic analyses that I've showed you earlier. And I'd like to say that this is kind of closing phase 2 of this research program, where phase 1 was learning how to sequence and assemble these genomes and do it well. Phase 2 was getting enough genome sequenced and starting to compare the genomes to find differences. And finally, we're starting to look at some of the more fascinating evolutionary questions that drive me. And so the work that I'm talking about here was done by Yi Liao, Xinwen Zhang, and Mehul Chakraborty in my lab. So if we move on to the next slide, what we're doing here is we're using Hi-C in order to measure the 3-dimensional structure of DNA. And so we're using this. So, as you might remember from molecular biology as a student, or if you actually study this, that the DNA is hierarchically organized into different three-dimensional structures, where you have DNA wrapped around histones to form nucleosomes, then chromatin loops form, and then individual loops can be variable and interact with each other. And as the DNA folds up into higher and higher orders of structures, you find these things called chromatin domains, or topologically associating domains, or TADs. And these topologically associating domains are these regions of the genome that have a lot of individual contact with one another, but are more or less distinct from adjacent regions of the genome because they have a they do not touch each other. So basically, if you're thinking about a big messy ball of yarn, it's which regions form the knots that are interacting with each other with little strings between them. Okay, so if we move on to the next slide, what we've done in order to assay this is pretty typical. We used actually a commercial approach to do this. Is we just you cross-link DNA, cut it with restriction enzymes, ligate the DNA to itself, and then free it, and then sequence it. So what you've done is you've taken DNA that used to be in physical contact in sort of 3D space, but not local contact in terms of linear. So they could be several base pairs away, or several thousand, or several 100,000 base pairs away, but they're at least touching each other in 3D space. And what we've done is frozen those 3D topological connections and sequenced. And anytime you see a contact between two disparate parts of DNA, you think they're in three-dimensional contact. And so if you do this over many different restriction enzymes, you can get a good map of the association of DNA with each other. For someone interested in genome assembly, this helps you scaffold the DNA and tell you the order and orientation of different contigs in the DNA assembly. But if you actually think about the pesky signal, which makes it hard to do scaffolding, those weird rectangles you'll see that are on the diagonal, that's actually biological information. That's knowledge about the three-dimensional structure of DNA in the cell. So if we go on to the next slide, if you, so just to remind you, so skip forward one more slide. And one more. And right now, we applied this approach to Drosophila pseudoobscura. Our initial idea was that we were going to do this in order to scaffold it. But we quickly determined that it was really important to also look at this three-dimensional structure of DNA, because it's a very important component of genome biology. So if we move to the next slide, first I want to remind you is that this is a classic result. We've put it into a new format here, but we've known this since back whenever Sturdivant was looking at different karyotypes of different species nearly 100 years ago. But the Drosophila chromosome arms are called Muller elements, and these elements are strongly conserved across the entire clade. However, they're very actively shuffled. And so you can see here on the right-hand side is Drosophila melanogaster, and the left-hand side is Drosophila pseudoobscura, and the connections are segments of homologous regions. And you'll notice that while each chromosome sort of connects to each other chromosome, they connect in a twisted, tangled mess, which just is indicative of a lot of reshuffling. So if you go to the next slide, so what we did is we took these connections and we compared the breakpoints of where we found those strings in the previous slide and shuffled the regions of TADs with respect to that. So randomly determine if TAD morphology and rearrangement breakpoints were independent, what would we see? And we expect the amount of conservation expected in green there, but the amount of conservation we actually see for TAD body conservation is in orange, and we see something similar for TAD boundaries, okay? And so these elements are strongly conserved and are very obviously conserved, and they're almost certainly functional. Now, we're not the first to see that, but we're using that for our studies in Drosophila pseudoobscura. So if we go on to the next slide and skip forward one more, now we are, let's go ahead and take, because a lot of work on TADs have been done in Drosophila melanogaster. So let's go ahead and take the TADs in Drosophila melanogaster and see how well they're conserved in Drosophila pseudoobscura, and we're comparing where the syntenic breakpoints fall with regard to the beginning of one of these topological domains. So it's a segment of DNA that interacts with itself, and you look for where the rearrangement breakpoints are, and you'll notice that the rearrangement breakpoints pile up near the boundaries of the TAD, and they're very rare inside the TAD itself, and they are not very high outside the TAD. Now, the reason why the outside of the TAD is a little bit higher than the inside of the TAD, and why does it pile up at the breakpoints? Well, we think that's because TADs are usually packed pretty tightly in the Drosophila genome, and so going too far away from the start, even if you're going outside the TAD, you're likely to find another TAD off in that direction. So what we think that happens is that it's okay to rearrange the genome near a TAD breakpoint, but you probably shouldn't rearrange the genome where it interrupts a body. So that tends to make things pile up at the boundaries, but not too far away from the boundaries, for fear of falling inside another TAD. So if we instead do the reverse experiment, if you go to the next slide, and look at TADs in Drosophila pseudoobscura and see how well they're conserved with regard to syntenic breakpoints in Drosophila melanogaster, you get the same pattern. So, and you can do this across TAD boundaries and TAD breakpoints, and you get similar results. So if we move on to the next slide, and this will be pretty close to the concluding paragraph. I asked, so we see this really strong conservation of topologically associating domain structure, and it tells you something important. So previously we've been thinking about duplicating genes or inserting a TE near a gene that changes its transcription, or things like that, things that change the structure and the meaning of coding elements. But another thing that becomes quite obvious when studying the 3D structure of DNA is that there's very strong conservation in how the DNA is organized. And so we asked this question, what does that do to mutations that affect the structure of TADs? So rather than using genes as the unit of interest, we're using TADs as a unit of interest. And so here what we've done is kind of the closest I can come right now to thinking about TAD, or to thinking about structural variation, polymorphism and divergence in a TAD context. And I'm really happy to think about better ways to look at this. But what we did is this odds ratio, and what we have on the top here is the divergence between, say, Drosophila melanogaster and Drosophila simulans in structural variation near TAD boundaries, right, versus the divergence far away from TAD boundaries, but not inside TADs. And we did the same thing with regard to polymorphism within Drosophila melanogaster. So we're doing the Simulins clade comparison versus polymorphism within melanogaster. And this is meant to evoke something similar to, the idea about maybe a McDonald-Kreitman table or something like that. It's not the same because, just simply not being near a TAD boundary is not the same as being synonymous and non-synonymous, but it's the best we can do now. And what you see here is that a large value of the odds ratio indicates that there's an excess of divergence at TAD boundaries relative to what you might predict from polymorphism if you're normalizing by the amount of TADs you see away from the boundaries. And if this were polymorphism and divergence for amino acid and silent sites, you would possibly interpret this as strong positive selection for amino acid substitution between species. But what we're seeing here is that tandem duplications have a very strong positive ratio where everything else is much, much lower, and usually quite close to 1. So, this we think is sort of interesting early peek at possibly the fact that duplications that are preserved between species are reinforcing these TAD boundaries where these insulator elements, I didn't have time to go into that, where they're also enriched for these insulator elements that sort of keep these compartments sort of transcriptionally independent from other compartments. And so we think that there might be some evidence that reinforcing those through copying the regions might actually be something that happens. And there's some other patterns that we can think about, but this is early days. So if we'll move on, I believe we can click down again, and we're coming to the end. First of all, I'd like to thank everyone who I acknowledged throughout the talk with pictures and papers. Please don't think that we did this all alone. Next slide. But also particularly the people in my lab, Mehul Chakrabarty, next. Luna, in my lab, who was my lab technician for a number of years, Edwin Solares, who is also in Brandon Gott's lab, who I believe also talks in this seminar series, and helped with the nanopore assembly. Xinwen Zhang, who's helped a lot with expression and paralog-specific duplication. And next slide. And Roy Zhao, who did the selection analysis for the cytochrome P450s. Next slide. And with that, I'll take questions.